This lesson is about how the information in DNA is used to build proteins. This is called the process of protein synthesis, and it occurs in a couple of different stages. We'll start first with an understanding of the central dogma or main idea of molecular genetics. The central dogma or accepted belief of molecular genetics is that DNA makes RNA, which is used to build proteins, which give us our traits. Also need to understand that DNA can be used to make more DNA, and so it's the genetic material. The process of DNA making more DNA is called replication. It occurs in the nucleus of eukaryotic cells, but in bacteria, prokaryotes that have no nucleus, it just occurs in the cytoplasm. The process of DNA making RNA is called transcription, and it occurs also in the nucleus in eukaryotic cells. The process of RNA making proteins is called translation. It occurs outside the nucleus in the cytoplasm. The process of transcription and translation combined is protein synthesis. However, even after the proteins are built, they can be modified, and information in RNA can be modified to give different proteins and different traits. So the process of RNA and proteins giving us our traits, we call that expression, gene expression. Now, in order to be a genetic material, there must be at least three conditions met. First, a genetic material must be complex. Life is a complex, the molecules are built out of a complex, so too simple of a molecule wouldn't have been able to be a genetic material. Second requirement is that this molecule or material be able to copy itself, and that occurs in DNA in the process of replication. The third, and probably most complex property of a genetic material, is that it must be able to control or direct other molecules. For this lesson, we're going to examine how DNA controls and directs other molecules, and it does that by building proteins. Here's a diagram of a cell nucleus, and outside the nucleus in the cytoplasm, you can see the nuclear membrane, the nuclear pore. We have chromosomes inside the nucleus, which is DNA wrapped around histone proteins, allow the DNA to coil and condense and protect the DNA from damage when it's not being used. Most of the time, DNA is not replicating, it's not copying itself. Instead, it's condensed into chromatin, which is the DNA wrapped around these histone proteins, more protected. But at times, the information in the DNA needs to be used to build proteins. So, depending on which protein needs to be built, that portion of the DNA needs to open up so that that message can be transcribed. So, in this example, we'll use the enzyme helicase to uncoil the DNA, untwist it from the double helix. The hydrogen bonds break between the complementary base pairs, and we're going to use single-stranded binding proteins to prevent the separated strands from reattaching to each other. So there it is, a single-stranded binding protein. Now, remember, DNA is always built in the 5' to 3' direction. So the top strand of DNA, we're going to use that information of DNA to build a similar molecule called RNA. It's important for the cells to differentiate between DNA and RNA because DNA is the master instructions. Very important. That's always kept in the nucleus, always protected. Imagine if damage were to happen to that. It's sort of like, imagine a set of blueprints being taken out to a construction site where they get run over by the tractor and spilled water on and caught on fire and they get damaged. They're irreplaceable. The original blueprints are never taken out to the work site. In the same way, the original DNA, our instructions, are never taken out into the cell to build molecules from. Instead, we take a copy, just like copies of the blueprints are made to take out to the work site. The original blueprints are kept inside a filing cabinet in an architect's office, since in the same way DNA is kept inside the nucleus inside chromosomes. But when we need the information, we uncoil the chromosome and untwist it, and then use the DNA to build RNA. Now we use the enzyme DNA polymerase to build more DNA. We're going to use a similar enzyme, Rna polymerase, to build RNA. Here's the RNA polymerase enzyme. It's going to travel this direction, 5' to 3', and as it does so, it's going to grab free RNA nucleotides that are floating in the cytoplasm inside the nucleus. In the nucleus there are free nucleotides, both DNA and RNA. Remember, the difference between RNA and DNA is that RNA uses uracil instead of thymine as a nitrogenous base, and that RNA has the sugar ribose instead of deoxyribose. However, both nucleotides are floating around, and its enzymes, the Rna polymerase enzyme, is the right shape to grab a hold of RNA nucleotides, incorporating them into the growing RNA strand. It'll match up complementary bases. So T matches up with A with U, C with G, until the entire RNA strand is built. Now at this point, only a certain section of DNA is uncoiled and allowed to open up. This section of DNA is called a gene. A gene is a section or a piece of DNA that codes for a protein or other product. In this case, I obviously have abbreviated. Real genes are somewhere in the order of a few thousand base pairs of DNA, a few thousand bases long to a few 100,000 bases long. Realize that these are very long molecules. Okay, once the RNA is built, it detaches from the DNA strand. The temporary hydrogen bonds holding it together break off, and it floats away from the DNA. The DNA recombines into the double helix again, leaving the RNA free in the nucleus. So here we see our RNA molecule freed from the DNA. The DNA is coiled back up into its double helix, which will further coil around the histone proteins and return to the chromosome until that gene is needed again. Now this first RNA that's built we're going to call primary transcript RNA. And in prokaryotic cells, this is used immediately, and it travels right away to the ribosomes where proteins start to be built. However, in eukaryote cells, certain portions of the RNA are not used to build proteins, and they're removed before the RNA can leave the nucleus. This is called the process of gene editing, RNA editing, or RNA splicing. And it happens inside the nucleus, where certain enzymes cut out certain pieces of the RNA. The pieces that were removed came from sections of the DNA that we're going to call introns. They're intervening sequences that are not used in the final product. The other sections of the DNA that are used to actually code for the protein are called exons. The RNA pieces from the exons are spliced together after the introns are removed, and we call this new processed RNA, we call it messenger RNA, or little m RNA, mRNA for short. The mRNA is tagged with chemicals at the front, a cap called a 5' cap, and then a tail made out of a bunch of repeating adenines called a poly-A tail. The cap is a modified guanine chemical, and is important in helping to differentiate where the beginning of the RNA is from the end. This chemical, this new messenger RNA, is now free to leave the nucleus and travel out into the cytoplasm, carrying the message from the DNA out to the ribosomes to build the proteins.